Hi everyone. Today we are going to continue from where we ended last time on the renal system. Okay, so allow me to start looking at what we call now the renal blood supply. Okay, so basically last time we're looking at the anatomy of the renal system and the importance of us learning renal physiology as medical students. Okay, so now renal blood supply, the two kidneys which we're basically talking about. Yeah, the two kidneys lie on the posterior wall of the abdomen outside the peritoneal cavity, which is known as the retroperitoneal. Each kidney receives blood through the renal arteries, both from the left. Each kidney receives blood through the renal arteries, both left and right from the descending aorta. Okay, so now the renal veins from the two kidneys empty blood into what we call the inferior vena cava, which we did discuss about in the anatomy. Then about the kidneys receives about 125 ml of blood per minute and about 180 liters in 24 hours. So this is just talking about how much blood goes to the kidneys every day. Okay, yeah, that's about 180 liters approximately in 24 hours. So this amounts to 1.1 to 1.3 liters per minute, which is just under about 25% of cardiac output. Basically, I'm sure you know what cardiac output is. So cardiac output simply basically means the amount of blood ejected from the ventricles per minute. Okay, that's known as cardiac output, and the formula is cardiac output is equal to heart rate multiplied by stroke volume. Now let's continue. So now the renal arteries enters the kidneys through what we call the ileum, and then branches to form what we call segmental arteries, which progressively divide into lobar arteries, interlobar arteries, arcuate arteries, and interlobular arteries, also called radial arteries. So now, when you're just looking at the text, it looks a bit complex and a bit difficult to understand. But when you look at the diagram, it will be very, very easy for you to understand. So you'll be able to picture it or know that the renal artery subdivides into what we call segmental arteries first, which later divide into what we call lobar arteries, then interlobar. So segmental, lobar, interlobar arteries, arcuate arteries, interlobular arteries, which are also called radial arteries. So the interlobular arteries are the ones now that divide or subdivide to form what we call afferent arterial, which supply each and every nephron in the kidney. Okay, so each of these divides into multiple capillary branches to form what we call the tuft of vessels in the glomerulus called the glomerular capillaries. That's where large amounts of fluid and solids, except the plasma proteins, are filtered to begin with urine formation. So we'll talk about how these are able to join up. I like diagrams because they're easier to explain and they're straightforward. So now, from what you're talking about on the previous slide, so you can be able to see that we have what we call the renal artery there, okay? So this renal artery you're able to see there is the one that is going to subdivide into what we call first the segmental arteries, then it will go into what we call the lobar arteries, then after that interlobar arteries, okay? So after the interlobar arteries, then we are going to go into what we call the arcuate artery. The arcuate artery is going to first subdivide into what we call interlobular artery, and then the interlobular artery are going to further subdivide into what we call a joint actually. This is a zoomed in image showing what we call the afferent arterial, as you can see, okay? So interlobular artery is there, okay? Yeah, so this interlobular artery, as you can see, okay, is the one that has been zoomed in. Then it subdivides, or it joins what we call an afferent arterial. And this afferent arterial is the one that joins what we call the nephron. Basically, as you know, we did discuss about what a nephron is, which is the basic functional unit of a kidney. So this diagram you're able to see here is known as a nephron, okay? Is known as the nephron. All right. No wonder if you look at the shape of the nephron, we haven't included the collecting tubules, hence because they're not part of the nephron. So what you're able to see is, the interlobular artery then joins into what we call the afferent arterial, and the afferent arterial is going to form a tuft of capillaries known as the glomerulus, which you're able to see there, in a cup known as the Bowman's capsule. Then after that, going to join to form what we call the efferent arterial, okay, which are the only arterioles that are formed by joining a bunch of capillaries, because mostly when capillaries join, they're supposed to form a vein, okay? But these special type of capillaries are going to form another arterial. They're coming from an arterial, then they're going to form another arterial, right? So this is the only one in the body. So now after this, they'll form what we call an afferent arterial, then exit the Bowman's capsule, and then further subdivide or branch off to form what we call the peritubular capillaries. Peritubular capillaries, okay? So these peritubular capillaries, again, will coil around the nephron, okay? Most precisely, they are found in the cortex, okay? Then below here, as you can see, these are a different type of blood capillaries that are found in the nephron, which are found below, or which are found in the medulla, which are known as the Vasa recta, or Vasa recta, okay? So this Vasa recta is a special type of capillaries that are found in the medulla, and they're responsible for what we call the countercurrent mechanism, which has got the countercurrent exchanger and the countercurrent multiplier. Then later on, they're going to join up to form what we call the venule. Then this venule will join what we call the interlobular vein. Then again, from the interlobular vein, arcuate vein, interlobular vein, after interlobular vein, arcuate vein, then interlobar vein, then it will go into what we call the lobar veins. Then after that, it will join to form what we call the renal veins. Hence, it will leave the kidney and join what we call the inferior vena cava and take the blood back to the heart for oxygenation and other processes. So basically, this just describes the anatomy, describes the secretory system of the kidney, all right? Yeah, so there will be a lot of text which will be talking about this, which basically well have already discussed. And yeah, so glomerular capillaries, they are the only capillaries in the body that drain into the arterioles, called efferent, like I said. So the efferent arterioles are short, straight, and the branches of the interlobular arteries forming glomerular capillaries in the Bowman's capsule. And the glomerular capillaries drain blood into the efferent arterioles, which has relatively little smooth muscles. Their main function is ultrafiltration, as you know, okay? So basically, this will talk about the things that I've already talked about, the Vasa recta and what it does, which we'll talk about later, okay? So here's just an example of the Vasa recta. So the Vasa recta is a portion of the peritubular capillary system which enters the medulla, where the physiological concentration is in the interstitium and is high, and it acts with the loop of Henle to concentrate the urine by a complex mechanism of countercurrent exchanger and using urea. If the Vasa recta does not exist, the high concentration of solute in the medullary interstitium would be washed out. Hence, the kidneys would be deemed to be non-functional. So now what you're seeing here, these are two different types of capillaries. Two different types of nephron. So the one that you are seeing here is known as the cortical nephron, and this cortical nephron is found in the cortex, and it has got no Vasa recta, right? And it has only got what you call the peritubular capillary. But what you are seeing is known as a juxtamedullary nephron, which has got two parts. It has got two types of blood capillaries. It has got the Vasa recta there, and the Vasa recta below there, and it has got what we call the peritubular capillary. So, like I said, the peritubular capillary is found in the cortex, while the Vasa recta is found deep down in the medulla, and is responsible for keeping the interstitium of the medulla to be concentrated. That's the purpose, and hence help to us to make either dilute or concentrated urine, depending on the conditions. All right, so now this is just trying to talk about the things that you're going to learn about under filtration, okay? So now in the nephron, you're going to find that to the left is a single generalized uniferous nephron, okay? Uniferous tubule. So more than 1 million of these tubules act together to form what we call urine. That's a function, and three main mechanisms happen. So there's what we call glomerular filtration, tubular reabsorption, and tubular secretion, okay? Yeah, and there will be also excretion, right? Yeah, amongst those. So two major parts. We have got a urine-forming nephron and the collecting duct, which concentrates urine by removing water from it, okay? Yeah. So these are just the stages that you can see. So remember, this is the interlobular arteries, as you remember, and this is known as the Bowman's capsule. Then there's also what we call the glomerular capillaries, right there, capillaries, which is found in the Bowman's capsule for ultrafiltration or pressure filtration. Then the efferent arterial, like we looked at, it in the blood capillary system of the kidney. Then it goes down the rest of the renal tube containing filtrate and peritubular capillary and to interlobular veins. So now this is just trying to talk about ABC. So ABC, when you look at the key, is showing glomerular filtration, B, tubular reabsorption, then C, tubular secretion. So these are still talking about the same two different types of nephrons, but this image is more refined and a bit more detailed to be able for you to appreciate. So if you look on this far left, you're going to see the cortex, okay? And you're going to see the medulla. So now there's the outer zone and the inner zone of the medulla. So on top there, if you can see on the cortical, just only a tip of the loop of vein just goes into the medulla, but most of it is found in the cortex, hence it's called cortical nephron, all right? Then when you look at this, only a small portion called the peritubular capillary is found in the cortex. Then the entire part of the nephron is found in the medulla, hence it's called the medullary nephron. So these are the two types. So this one, remember, it has got the Vasa recta and is responsible for diluting and concentrating urine. So as the glomerular filtration enters the renal tubule, it flows sequentially through the successful parts of the tubule, the proximal tubule, the loop of Henle, the distal tubule, the collecting tubule, and finally the collecting duct before it is excreted as urine. So substances are selectively reabsorbed from the tubules back into the blood, and others are secreted from the body into the tubular lumen. So that's what this is about. So what you're seeing on the screen now is known as a nephron, okay? So this nephron has got different parts, and you're supposed to know all these. So now, as you say, we've been talking about the afferent arterial, okay, which further subdivides into what we call the glomerulus, and it exits via what we call efferent arterial. And I believe we went through the circulatory system of the kidney. Then what you are seeing here is known as the Bowman's capsule. And the Bowman's capsule extends to what we call the proximal tubule. So there's what we call the proximal convoluted tubule. Then after that, there's what we call the thick descending segment. Then there's what we call the thin descending segment. Then there's a hairpin bend. Then as you start going up, we've got what we call the thin ascending segment. Then there's a thick ascending segment. Then there's what we call the distal convoluted tubule. All right, this is what we call a distal convoluted tubule. And as you start going there, it's going to be called the collecting duct. So this diagram, I think you've already gone through it, but the only thing that I can put an emphasis to, if you look at this equation here, is excretion. So excretion is equal to filtration, reabsorption plus secretion. So there's filtration happening there. There's ultra or pressure filtration, or what we call happening there. So everything goes into the Bowman's capsule. Then minus the reabsorption plus secretion. So what is left is known as urine, right? That's known as excretion. So basically you follow both ones, yeah, if you want to come up with that. So now these are what we call specialized cells found in the nephron. So as you can see in the glomerulus there, then after that we've got the proximal convoluted tubule. In the proximal convoluted tubule, what type of cells are found there? There's what we call proximal tubule. Okay, in the proximal tubule, there's what we call brush border. Proximal convoluted tubule, there's the mitochondria there, right? And there's what we call thin descending limb of loop of Henle. So here we have got brush border type of cells, okay, in the proximal convoluted tubule. And if you look at the mitochondria, there's plenty of mitochondria, meaning there's a lot of active transport happening there, all right? Yeah. Then after that, there's what we call the thick ascending limb of the loop of Henle. As you can see, it has got also some cells, which has got a lot of mitochondria. Then there's also what we call the distal convoluted tubule, which has got also cells with plenty of mitochondria. And then, if you look at these two types of cells, the one that's found here and the one that's found here, meaning there's no active transport. There is active transport. There is active transport. Here there's less mitochondria, meaning there's less or no active transport. Okay, yeah. So descending limb, ascending limb, and this is the loop of Henle. Yeah. Then here we have got what we call in the distal convoluted tubule. I mean, as we are doing the distal convoluted tubule, going into what we call the collecting tubule, we have got what we call the intercalated cells, and we have got what we call the principal cells. So these two types of cells that are responsible for what we call acid-base balance. Then there's also what we call the medullary collecting tubule, which are found in here. And these cells have got what we call aquaporins, and aquaporins are responsible for absorption of water, all right, due to certain conditions. When you find vasopressin attaches to this same type of cells, they're able to allow water to flow into these cells. All right, now urine that is formed, and all the substance in the urine represents the sum of three basic renal processes using glomerular filtration, tubular reabsorption, and tubular secretion. There's also what we call the renal tubular reabsorption and secretion. This diagram, I think We have already talked about what is there, and there's a better diagram which you can be able to appreciate. So now there's also what we call the renal corpuscle and the filtration membrane. So now this is just trying to talk about the cells that are found there in the renal corpuscle and the filtration membrane. So the renal corpuscle has to do with different types of cells that are found there. So if you look at it, you've got afferent artery, afferent artery, this is the glomerulus, and the cells lining the Bowman's capsule. There's what you call the vascular layer. You find that if you go on YouTube, you're going to find that there are a lot of videos concerning this. So there is this part. So if you see this part where it's reddish, this is where we have got blood capillaries of blood. So in between you have got cells, because you say blood capillaries have got spaces in between, she's able to allow substances to squeeze through. So now, if you look at this, you're going to find that there's what we call fenestrations, and there's blood, and there's also what we call capillary endothelium, and there's also what we call the basement membrane. So here is the filtrate, then there's the slit, there's what we call slit pore, and there's also what we call podocytes. So podocytes are cells that are found on the other side which are found outside of the Bowman's capsule, which we'll talk about later. So here there are slits there, and there are also slits there. So if you go in detail, you are going to find that there are about 3 layers which substances need to cross through what we call after ultrafiltration. Okay, so on the capillary side, there's the basement membrane, which has got also charges, which is able to repel certain substances, and there's also what we call podocytes, which also have got slits. So one thing you're supposed to understand is that under pressure filtration, all substances are able to be squeezed into the Bowman's capsule, except proteins, or you can say globular proteins, such as albumin. They can never go into the Bowman's capsule. All right. One is called filtration, because other substances are going to be prevented to go into that. So now this is another diagram trying to show the glomerular capillaries and their podocytes. So if you can see this, this is the pedicels, then there's a podocyte cell there, then there's a capillary lumen there, there's a filtration membrane, as you can see. In between, then there's a basement membrane there, and there's also fenestrated. So fenestration has to do with just that these are blood vessels that have got pores or spaces in between, all right? Yeah, these capillaries just got spaces in between, as fenestrated capillaries. Okay, and then there's also what you call the capillary endothelium, as you can see there, and there's also filtration slits, which are found on the other side, which are able to allow substances to go through. So this diagram is just trying to show substances that can be able to go through the filtration mechanism on the renal corpuscle. Okay, as you saw, you're going to find that. Let's look at filtration membrane. So there's glomerular filter. So the substances that have managed to go through the renal corpuscle and go into the Bowman's capsule is known as the glomerular filtrate or renal filtrate. So this glomerular filtrate is what you're going to talk about, some of the substances that are found, some of them. So now, here is the membrane that we already talked about. So if you see, this is the side where you put the blood capillaries. So in the blood capillaries, you know, there are 2 types of cells, okay? Okay, yeah, I mean, sorry, there are 2 types of, there's what we call white blood cells, and there's also what we call red blood cells, and there's also rod-like shaped structures, which are known as platelets. So now, this is what we call small proteins, large proteins, filtered here, all right, as you go. So you're going to find that filtrate includes water, glucose, amino acids, ions, urea, and many hormones, vitamins B and C, ketones, and very small amounts of protein. So all these are able to go through. So remember, they're saying small proteins. Please don't misquote me. Small proteins cannot, can go through, but large proteins can never go through. Yeah, because of this type of filtration mechanism. Remember, there's a filtration mechanism on the blood capillary, there's the basement membrane, there's another filtration mechanism on the other side of the Bowman's capsule. So it's very difficult for these proteins to be able to go through. Yeah, so this diagram is just trying to talk about what we call the juxtaglomerular apparatus. So the juxtaglomerular apparatus, what you're supposed to know is juxta means near, okay, or next to. So when you talk about juxta, this is a complex or juxtaglomerular apparatus involves what we call the macular denser cells and also the cells which are found at the renal corpuscle. So let's just go through the text. So what is the juxtaglomerular apparatus? Answer: juxtaglomerular apparatus is specialized organs situated near the glomerulus of each nephron. Juxta means near. So number 2, show the 3 types of cells of the JGA. A. So juxtaglomerular apparatus, and state their function. Okay, it's a question. So now the juxtaglomerular cells or JG cells are modified smooth cells of the afferent arteriole that secrete renin, a proteolytic enzyme. Then there's also what we call the macular denser cells, are located in the walls of the terminal end of the thick ascending limb of Henle. Okay, so the distal straight tubule that monitor a decrease in the concentration of the sodium in the distal tubule. So now let's do this. So now when you look at these two things, so it's involving the juxtaglomerular cells, which are found in the afferent arteriole, and also it involves what we call the macular denser cells. So these two produce different types of substances, but they are very important when it comes to what we call the RAAS system, which is known as the RAAS system, renin angiotensin aldosterone system, all right, which we'll talk about later as we go on. Yeah. So now the juxtaglomerular apparatus. So what you are able to see here is known as the glomerulus, the glomerular capsule, and region of the juxtaglomerular nephron. Okay, this yellowish part of the nephron. So this is not a glomerulus, etc., etc. Then when you get this portion, then you zoom it in. So as you can see, we have a distal tubule there, and there's the afferent arteriole and the efferent arteriole. So this is known as the juxtaglomerular apparatus, and it's very important when it comes to the RAAS system. So this is known as the glomerulus. So if you can see, is the afferent arteriole coming in, and this efferent arteriole. Then what you are able to see is known as the granular cells and macular denser type of cells. So this is basically called the juxtaglomerular apparatus. All right. So that's where now we come in and talk about what we call the RAAS system. Basically, what is the RAAS system? The renin angiotensin aldosterone system. Yeah. So this is just basically talking about the same thing that we're just from looking at. Yeah. Granular messenger cells. So here we're just trying to talk about the messenger cells that are found at the renal corpuscle inside the Bowman's capsule. So these are the different types of cells and the nerve fibers that are connected, the JG cell. So basically all what is there, we already discussed it and we can't go in detail. But if you have any questions, please let me know. Yeah. So now, this is just trying to show the RAAS system, okay? The RAAS system basically talks about what happens when your blood pressure is not normal, okay? Yeah, so for example, it has to do with what can cause you to get your blood pressure back to normal, or to increase your blood pressure. For example, in case you are dehydrated, in case you have got a condition that leads to you having a low blood pressure. So this diagram talks about what you call the RAAS system. So now, when you look at this, RAAS, so now stimuli. So now when you look at this RAAS system, so here there's a liver. So we have to start from here is the inside the kidneys. Remember we talked about the juxtaglomerular apparatus, right? So the juxtaglomerular apparatus, what is it and what does it do? That's what we need to know, and that's what we need to appreciate. So now when you look at this diagram, you're going to find that inside the afferent arteriole, there are cells known as juxtaglomerular cells. So these juxtaglomerular cells are specialized cells that produce what we call pro-renin, which is going to be activated later on to what we call renin. So now what stimulates the secretion of renin? It's when the blood flowing through the afferent arteriole. So the juxtaglomerular cells, they're more like baroreceptors. They detect pressure difference. So as blood is passing through there, they detect that the pressure is very low. It's going to cause the secretion of what we call pro-renin. It's going to be going to what we call renin. So once renin is produced, the renin is going to go and enter into circulation. So once it enters into the circulation, so as you can, then this enzyme is going to enter into circulation. Then the liver is also going to produce something which is known as angiotensinogen, right? So angiotensinogen is a protein that's going to be produced in blood. So that angiotensinogen plus the renin which is produced by the juxtaglomerular cells are going to react, as you can see in this flow chart, to produce what we call angiotensin 1. Okay. So once angiotensin 1 is produced, then this angiotensin will continue to circulate into the blood and to go to where we call the lungs. So in the lungs, there are what we call endothelial cells there that are capable of producing an enzyme which is known as angiotensin converting enzyme. So those endothelial cells of the lungs, when they produce this enzyme known as angiotensin converting enzyme, the angiotensin 1 will be converted. If you can see here, these endothelial cells of the blood capillaries, is going to be angiotensin 1 will be converted to what we call angiotensin 2. What is responsible for that is ACE, angiotensin converting enzyme. So once you convert it from angiotensin 1 to angiotensin 2, which is more reactive, this angiotensin 2 will continue to move into the blood, and it's going to go to various areas of the body, okay? Number one, it can either go to the heart and influence the cardiovascular system, okay? Or let me say, if you go to the cardiovascular system and influence it, hence causing what we call vasoconstriction. If you know, if the blood vessels reduce in volume, hence the pressure is going to increase. An example that I can give for this one, if when you're altering the garden using a hose pipe, when you try to reduce the size of the lumen of the hose pipe, the pressure increases. So that's the same phenomenon it uses. Then the other thing is that it will go to the adrenal cortex. It will go to the suprarenal glands. The suprarenal glands are going to produce what we call aldosterone, okay? So aldosterone is a hormone that is responsible for going into affecting the distal tubule, hence making it to absorb a lot of salt, hence what I mean by absorbing a lot of sodium. So where sodium goes, water also follows. So it will be moving from the proximal tubule back into the blood, and water will also follow, hence increasing the volume of water. So if you increase the volume of water, even if you talk about a tap, you're using your pipe watering, then you increase the volume, you open the tap more, the volume of water going through will increase, hence the pressure. So it's the same phenomenon. Then again, it will go to the same, it will go to the brain, okay? Yeah, so in the brain, there's an area called what we call the pituitary, right? So there's what we call posterior and anterior. And the posterior pituitary has got one function of producing what we call vasopressin, or ADH, or antidiuretic hormone. So the ADH will be produced and it will be sent to the kidneys, to be more precise, to the collecting tubule and stimulate what we call the cells that contains aquaporins, to allow a lot of water to be flowing from the collecting tubule from urine going to the bloodstream, hence also increasing the volume of blood. All right. So these are some of the mechanisms that lead to increasing the blood pressure. And then the last one has to do with thirst. It's going to go into the thirst, it's going to go and stimulate the thirst receptors and allow you to drink a lot of water, hence increasing the volume of blood. So the phenomenon about the RAAS system is to increase the blood pressure. So today we are going to end here until next time. Thank you very much, and God bless.